In this screencast, we're going to focus on the structure of DNA, but I'll also briefly touch on something called the central dogma. In class, you are going to realize that scientists worked a lot like detectives, and they spent a lot of time looking at the structure of DNA, and it was really important for them to understand, how DNA is structured. And so there are people named Griffith and Avery who are really important in discovering that structure, but we'll focus more on them in class. We know that DNA carries genetic information, but explaining how it can do this is a little bit more complicated. The DNA has to assemble proteins, but then the genes have to replicate and be inherited as well, which are complex things that we'll talk about over the course of the term. So DNA is a pretty special molecule, and it has a very special structure to it. DNA stands for deoxyribonucleic acid, with a D and an A underlined there for you. And it's a really unique molecule. So it's actually, of the big four, a nucleic acid. And it's made up of nucleotides, I'll go on and identify what that is for you in just a minute, that are joined into really long strands or chains through covalent bonds. You may remember this from other classes, or you may remember it from the fall term, but nucleic acids are long and they tend to be slightly acidic, that are originally identified in the nucleus of the cell. Nucleic acids are a macromolecule, right? We have those four biomolecules, and nucleic acids are one of them. And they're made up of these smaller subunits that will be linked together, and they form these long chains. And the nucleotides are these building blocks of the nucleic acids. Each nucleotide has three basic components to them. First, they have a sugar, which is a 5-carbon sugar. It's called deoxyribose. Then it also has a phosphate group, and the final component is a nitrogenous base. These are, I guess simply put, just bases that contain nitrogen. And DNA specifically has 4 kinds of these nitrogenous bases, which are adenine, guanine, thymine, and cytosine. And when we're talking about them, we often just abbreviate them by their first letters. So A, G, C, and T. So the nucleotide that we have made up of the deoxyribose, the phosphate, and then nitrogenous bases, that creates a strand of DNA when you combine a bunch of them, and they're joined by covalent bonds that will be formed between the sugar of one nucleotide and the phosphate group of the next. So let me show you what that looks like. There is our 5-carbon ring, our deoxyribose. Then coming off of it, we're going to have a phosphate group. And then coming off of the other side, we'll have either our adenine, guanine, cytosine, or thymine. So I'll abbreviate that as NB for nitrogenous base. So if we want this nucleotide to bond with another to start forming a DNA chain, we're going to have that phosphate group of that second nucleotide have a covalent bond with the one above it. So we see that our nitrogenous bases there are sort of sticking out sideways from that nucleotide chain. And we can have the nucleotides joined together really in any order or any sort of sequence. And the bases, just so you know, have a chemical structure that's going to make them really good at absorbing UV light. And we can actually determine the amount of DNA in a solution by looking at the amount of light it absorbs at a certain wavelength, like 260 nanometers, which is the UV region where we have the electromagnetic spectrum. Okay, so here on this slide you can see the general structure of DNA. Keep in mind, DNA is our universal code for life. It enables organisms to transmit hereditary information, and also, along with the environment, will help to determine the organism's characteristics. If we zoom out a bit and look at DNA, it's often shown in this general shape, which is called a double helix. Again, we'll learn more about what that means later on. But this is just sort of showing the rungs of the ladder. I haven't shown the nitrogenous bases in between. You'll also notice from this drawing, and obviously this isn't the best since the lengths aren't exactly the same, but we see that every green is always going to match up with a blue, and all of the reds there will match up with the lighter turquoise blue. And that means that our nitrogenous bases, or nucleotides, tend to bond to specific other nucleotides, nitrogenous bases, that have a higher affinity for them. So knowing that DNA is made of these, long chains of nucleotides, it's only really the beginning of understanding DNA. And so the next steps were discovered by someone named Chargaff, and also someone named Rosalind Franklin, and Watson and Crick. Again, we'll learn more about this next week in terms of its general double helix-like shape. Do keep in mind, though, that we have specific base pairings that happen, and again, more on this with Watson and Crick when we get there. But we find that there are hydrogen bonds here between our nitrogenous bases along the center. So these are where we have our hydrogen bonds, versus over here on the rungs of the ladder, these are covalent bonds along there. But the bonds here in the center, those hydrogen bonds, are only happening with certain base pairs, and that is adenine with thymine, and vice versa. So the thymine will bond with adenine, and our cytosine with guanine, and the opposite there again, guanine with cytosine. So this nearly perfect fit that we have between A and T and G's and C's, those nucleotides, it's known as something called base pairing. Back at the beginning, I told you I also was going to tell you about something called the central dogma. And this is an idea in molecular biology, and it describes the way that we do two processes called transcription and translation. And this is the way that information in genes will flow into proteins. It states that DNA will be transcribed into RNA, and then that RNA will be translated into protein. So transcription is just going to be the synthesis or the making of an RNA copy of a segment of DNA. And then we have RNA, this is synthesized by an enzyme that's called RNA polymerase. So that's just a general idea, and we will go into the specifics of transcription and translation later on in the term. Okay, that's all I have for you here on the beginning of DNA structure, and a little bit on the central dogma. Again, more on transcription, translation, and more specifics on the double helix shape that DNA has in class.